As I said, we will now study the effects of radiation at the chromosomal level. From the DNA, now we move a little bit up to the chromosomal level. So this is the human karyotype. I think you have already seen it. Just to remember, we have in our somatic cells, we have 46 chromosomes in all the somatic cells, 22 pairs of autosomes, and two sex chromosomes. Now, this is the normal karyotype. It's called the karyotype of the humans. So all of us have these chromosomes. But of course, the topic of this lecture will be the aberrations in this karyotype, the induction of this aberration and their consequences. So generally speaking, the aberrations can be divided in constitutional and acquired. Of course, constitutional are those that are already at birth, while acquired are those that come later when you have differentiated cells, so in the adult organism. Then they can be homogeneous or mosaic. You see, the homogeneous aberrations means that every cell in the organism has the same aberration. So this means that the anomaly must occur here at this early level of development. While they are mosaic, if they occur later, you know, when the embryo is starting to become a fetus, and then at that point you have an anomaly, so it will be mosaic. Not all of them will be not all the cells will have the same. Of course, mosaic is also if it is acquired. Not all the cells will have the same. Then they can also be numerical or structural, so in the number of chromosomes or in the structure of chromosomes, can be chromosome or chromatid type, depending on whether affect both chromatid at the same site, or only one chromatid and the other one is unaffected, and can be symmetrical or asymmetrical. This we will see, depending on whether the result of the aberration are two identical chromosomes or they are different. Generally speaking, you have to remember that when we talk about radiation-induced aberrations in adults, let's say, not, of course, in principle, you can also irradiate an egg. Then if you irradiate an egg or an oocyte, then in that case, you can have a constitutional homogeneous anomaly. But if you irradiate an adult, you will have all of them are acquired and mosaic. Now, how do we score the chromosomal aberrations? This will be the first. It's actually, this is very important. Here we have to talk about the methods, because what you score, depending on what you, how you paint your chromosomes, you will score. You will see different things. So essentially, you can see the chromosomes either in mitosis, which is the typical, historically, and it's more than 1 century that people look at the chromosomes in mitosis when they are thick and big. They are, you remember in the biology lecture, they are so wrapped about many histones that they become so big in the order of 2-3 microns and 0.2, 0.4 microns in diameter, that you can easily see them at the optical microscope. In interface, you generally don't see the chromosomes. They are there, you remember, but they are dispersed in the nucleus. They occupy large domains, but they are dispersed. However, you can prematurely condense them, PCC, prematurely condensed chromosome. We saw that you can do it if you kind of cheat on the cell. You make the cell believe that it's going into mitosis, so they will condense the chromosome. And that's the case here. You see, these are mitotic chromosomes with 2 chromatids per each chromosome. These are also mitotic chromosomes here, but these are instead, these are PCC. You see, in fact, there are only 1 strand. They look like little spaghetti, you see. actually the way that these PCC were induced was actually by fusion of a cell with, in mitosis, this is actually a hamster cell, with the human cell, a lymphocyte in G0. Because the mitotic cell has a large amount of mitotic promoting factor, the MPF protein, which is the protein that induces chromosome condensation, if you fuse it, this mitotic promoting factor will go into the other cell, the human cell, and will condense the chromosomes that are, however, in G0 and G1. So each chromosome is only one chromatid, not two chromatids. Of course, the second chromatids can only come after the S phase. These pictures here are all done in the so-called solid staining. So essentially, you stain with the paint, which is normally this is Gimsa, which is blue generally in color. You have to use the DNA is acid, so you have to use essentially basic liquid that binds with the acid DNA. But you can do better. You can do bending, and of course you can do fish. So we will see how much you learn if you do bending and if you do fish, and how many different types of fish fluorescence in situ hybridization is fish you can do. So Gimsa, as I said, is the simplest way to look at the chromosomes. You just stain them, you go to the microscope and you look at them. These are mitotic chromosomes after irradiation. And in this picture, you see a lot of aberrations. You see, the chromosome should look like this in mitosis. You know, should look like two chromatids connected by a centromere. Some of them should be smaller. Some of them, like this one, should be acrocentric, because if you remember, these chromosomes here, chromosome 13, 14, and 15 are acrocentric. Some are metacentrics, some are submetacentrics, some are really acrocentrics, 13, 14, 15, and also the small one, 21 and 22. So you see some of them here, but you also see very strange things. You see minute dots, fragments here, large acentric fragments without centromeres, abnormal monocentric, very long chromosomes with the centromere here, dicentrics, you see a chromosome that has two centromeres, tricentrics here, 1, 2, 3 chromosome, three centromeres in the same chromosome, chromatid type exchange. You see this exchange here, this aberration here, is not involving the two chromatids together, only one chromatid going one direction and the other going in the other direction. So many centric rings. This is a ring, is a chromosome that become a ring. Many different kinds of aberrations. It's a fascinating world already here in Gimsa. We will try to understand how can these aberrations be created. You can learn much more on the breakpoints of the chromosomes if you use G bending. This is a very old technique. It's based essentially on the trypsin, which is an enzyme, that digests the proteins along the chromosome, and of course will digest more the euchromatic part than the heterochromatic part, because the heterochromatic part are more thick than the euchromatic parts. So the result is a pattern of black and white bands that is very characteristic of every chromosome. You see, it is a full karyotype with the bending. And of course, if there is an aberration, you should be able to match exactly the bend in the right point. But that's, you immediately realize, is extremely difficult because it's only a black and white pattern. So to be able to distinguish, to say, okay, this is the point, you really have to do an enormous work. The world changed completely in the 80s, so approximately 1986, when in Livermore, I was actually personally, I was one of the first to use fish with the chromosomes, not because I invented the technique, but because in 91, I was in Berkeley for my PhD, and Berkeley is close to Livermore. So we got the first probes for fish from Livermore, where this technique had been invented. So how does it work? In principle, it's simple. No, this is, for example, let's say that you want to make a fluorescent staining of a portion of a chromosome. You can do the full chromosome, but let's say that you only want to see a gene, for example, or a telomere or something like that. Then what you need the sequence of what you want to see, and you need to label your sequence with the fluorescent marker, which is easy to do. So you label with the fluorescent marker. You only need a polymerase that takes the bases that are tagged with the fluorescent marker. So at this point, you have in a tube, you have a probe, which is fluorescent, and on the slide, you have the chromosomes that are not fluorescent. Now you have to denature both. And this you can do easily with heating. You go to high temperature in formaldehyde, you open the DNA of both the probe and the chromosome on the slide. Then you take your probe, denature it, and you put it on the slide, and you close with the cover slip. At that point, the denatured probe randomly will fall and will end up binding strongly the complementary sequence on the chromosome. Then you wash again at high temperature, so you make sure that all the non-specific binding, there will be a lot of non-specific binding, you can imagine, now small sequences are removed, and at the end you will have a beautiful image of the gene that you want to stain, of the area that you want to stain. Or if you have a full chromosome chopped in pieces and every piece is fluorescent and you hybridize, you will see the full chromosome fluorescent. That's what is done. These are old pictures, actually, especially this one. You see, this is a very old picture where two chromosomes were painted. This is one of the first pictures I got in Berkeley. Two chromosomes were painted. This is the chromosome 2 and the chromosome 4. The labels were in green, but the full slide was painted with propylium iodide, which is red. So red plus green give a yellow color in the image of the chromosome. And here, you see here something strange. You see a translocation. Here you see a yellow-orange chromosome. So here something happened, you see? You have the chromosome was broken, and instead there was a DNA double-strand break, if you want in modern terms, and instead of being properly repaired, it was misrepaired and rejoined to another chromosome. You see, you can do better, of course. So this is now 3 chromosomes are painted here. One is in green, one is in yellow, and one is in red, and all the others are painted in blue. This is an interface cells where, of course, you don't see the chromosomes, but you see the domains. As I told you last time, the chromosomes are there. You don't see them nicely like here, but they are there. And at this point, you can see wonderful aberrations. Look here, you have 2 chromosomes stained, chromosome 2 and chromosome 4. Chromosome 2 is in green and chromosome 2 is in red and chromosome 4 is in green here. And you see, this is a normal chromosome, this is a normal chromosome, but you see the other chromosome 2 has been broken in many pieces. One piece is rejoined to another unknown chromosome. This one is rejoined to another piece of the chromosome 4. This is also rejoined, green, and then there is another piece here. So now you start to see how with fish, you can see the complexity of the chromosomal aberrations that we had actually already seen in Gibbs. You can stain almost everything in fish. I told you, depends on which probe you want to take. For example, you can stain the different arms of the same chromosome with 2 different colors. This is called arm-specific fish. You see, so this is one chromosome, but the two arms are called the P and Q. P is the short arm, and Q is the long arm. In some cases, they have the same length. Chromosome 1 is essentially metacentric. but in many cases the P arm is short and the Q arm is long, and you can paint them in different colors. Or you can paint only the centromeres. You see only the centromeres of are painted here. These are centromeres of 2 chromosomes, and they are painted in green and in red. They should be 2 green and 2 red, right? So like in this cell here, this is now an interface cell, so no chromosomes are visible, but you paint with the centromere probe, and you see the 2 green and 2 red signal. But look at this one. You see an aneuploidy. These cells is aneuploid. There are more chromosomes. For example, these days, if you want to have a quick check on the trisomy 21, you know, that is the Down syndrome, which is the most common chromosomal anomaly, you can do like that. You just take the cells from the placenta, or now these days, even from the blood of the mother, you can isolate the few cells of the baby. You just paint it with the centromere of the chromosome 21. If you have 2 signals, it's normal. If you have 3 signals, the trisomy 21, it's a Down syndrome baby. You can paint the telomeres, as you see here. Here the telomeres are painted, and here is also how you can measure the length of the telomeres without. You can do it in molecular biology, of course. You can cut the telomeres, you can measure the number of bases with electrophoresis, but you can also do it physically if you want, measuring the intensity of the fluorescence of the telomeres. This is proportional to the length of the telomeres. And you see that in some chromosomes, the telomeres are a little bit longer than in other chromosomes, already normally. Finally, you can do the best, I mean, the most spectacular thing. You can play with the colors in a combinatorial way, so that at the end you can stain all the chromosomes, all the 23 chromosomes, if you want, with different colors. This is done by the computer, of course. It's a combination of different colors. Then the computer overlaps them, and you can build these beautiful karyotypes where you see every chromosome has different colors. This is actually my own karyotype from my blood. So these are chromosomes from my blood. And so you see all the pairs of autosomes, 22 pairs of autosomes, and the 2 sex chromosomes, the X, which is obviously of maternal origin, and the Y, which is obviously of paternal origin. Which one of these 2 is the mother and the father, we don't know, because of course they don't look different, but for this one we are sure. The sides, as you have seen in the cartoon, now here is more clear. The sides, you see, is essentially decreasing, not strictly, but pretty much decreasing, going from the low number to the high numbers. You see 21 and 22 are very small. Y is also pretty small. 1, 2, 3, sorry. 1, 2, 3, 4 and 5, you saw, are pretty big, and you see that 1 and 3 are almost metacentric, while, for example, 4 and 5 are clearly submetacentric. Now, instead of using these 23 colors to paint 23 different chromosomes, I can say, okay, you know what? I can take the 23 chromosome and paint only 1 chromosome. If I do that, I get something like that. So this is called M-bend, multicolor bending, and this somehow is similar to the G-bending, but it's much better because now each bend has a different colors. There is no correlation between the bends and the heterochromatin and euchromatin as for the G-bending. This is only a sequence issue. But you see, in this chromosome, you have 23 bends in different colors, and you can now see aberrations that you will not be able to see in M-fish, because you understand that M-fish is ideal if you have exchange between different chromosomes. But if you have an aberration within the chromosome, you can hardly see it because it's always the same color. Look at this picture, for example. This is how it looks like under the microscope. Apparently, a big mess. Then the computer makes its magic, and that's now how it looks like. So this is a normal, this is a chromosome number 5. This is a normal chromosome number 5, but the other chromosome number 5 has been severely broken in 3 pieces. You see 1, 2 bound to this chromosome and 3 inserted into this other chromosome. And I can know where. I can now bend, I can check the breakpoints in and bend. Can we mix? Can we try to mix M-fish and M-bend? Well, one attempt to mix is to do what is called the rainbow cross-species fluorescence in situ hybridization, or to make it simple, RX fish. Rainbow cross-species fluorescence in situ hybridization. What do I do here? I take human chromosome, and now I hybridize the human chromosome with a gibbon DNA. So this is a gibbon. Now the gibbon is very similar to human, so the hybridization is pretty good, but there are many translocations, if you want. I mean, it's not, the position is not the same. So what happens is that I see some bending, you see, in the chromosome. These are chromosome 1, chromosome 2, chromosome 3. Not as many bends as I used to see with M bending, but some bends. So I can now see, for example, in this specific karyotype, there is a translocation. You see, there is which is shown here. The karyotype is 46. There are 46 chromosomes. XY, you see X and Y. And there is a translocation between the chromosome 14 and the chromosome 17. This translocation 14, 17 is shown here. This piece here are the 2 small pieces that were broken and are rejoined. So together they form a reciprocal translocation. We will see it in a way. But you see that, let's say, using RX fish, I can kind of mix the M fish with the M bend. The resolution is not as good as M bend, and the images are not as clear as in M fish, because now I have different colors along the chromosomes. But you can have a lot of insight. You can learn a lot with this technique. Another way you can do arm-specific M-fish. So now you can again do the M-fish, but the 2 arms of the chromosome have different intensity, so they look different, you see. With arm-specific M-fish, you can see, for example, exchanges between the 2 different arms that you will not see if you use normal M-fish. This is an example. You see, these are different exchanges where you can now see this is a very complex exchanges here. And just to show you, where you can actually, these are actually pericentric inversion. We will see the pericentric inversion later, where you can see how useful can be arm-specific M-fish in determining the type of aberration. And this is another example. You see where we have, this is apparently is a dicentric between chromosome 5 and chromosome 3, chromosome 5 and chromosome 3. But this is the P arm of chromosome 3. This is the Q arm of chromosome 3, P arm of chromosome 5, Q arm of chromosome 5. So you see, and that's how the dicentric looks like. Okay. In the next part, we will start now to classify the radiation-induced chromosomal aberrations.